morphology, the body morphology, behavior, and the lipid metabolism. So, this will be my presentation outline. We'll see a little bit of introduction, objectives, materials and methods, results and discussion, and then we have some conclusion. And if there is any further questions, maybe it will be welcome. My main objective in this study was to assess the effect of ciprofloxacin on the body morphology of C. elegans. The second, it was to check the impact of ciprofloxacin on the PD metabolism. And the final objective was just to observe to assess the behavior toxicity of ciprofloxacin in C. elegans. Introduction. So we say ciprofloxacin is a fluoroquinolone antibiotics which is used which is prescribed for the treatment of various bacterial infection. And one of its major concern is ciprofloxacin. It has pseudopersistence. The pseudopersistence, we say, the phenomena where there is continuous emission and degradation. Once it gets into the environment, its degradation is in the emission is just continuous within the environment. So you find it takes a long time into the environment without being degraded. So this has been a problem of concern with many antibiotics. One of them being ciprofloxacin. So even if it is found at its low concentration within the environment, for example, in the water matrix or soil or whatever, it may cause a substantial environmental distress to different organisms, including aquatic organisms or soil microbes, etc. There are different studies which has shown, previous studies which has shown like the effects of this ciprofloxacin to untargeted organisms. These untargeted organisms can be zebrafish, nematodes, fish, and earthworms, etc. So we say the toxicological impact of ciprofloxacin, it occurs in different organisms through different mechanisms. So once this ciprofloxacin is within the maybe water matrix or soil, it can cause different distress to untargeted organisms through different mechanisms. So this mechanism can be generation of free radicals, which can create apoptosis, induction, and the structural degradation of DNA. Direct on the protein synthesis, some will act on the cell wall or cell membrane. So specifically, this ciprofloxacin, they just interfere with the DNA replication or by just interfering with the enzymes. So once this ciprofloxacin goes to untargeted organisms, it tries to impose the same effect to the cell of untargeted organisms, which are found around the environment. And hence, its toxicological effects starts to be observed within that particular organism. Materials and the methods. So the main materials we used in our study was ciprofloxacin. This is an antibiotic which is used for the treatment of different bacterial infection, especially both gram-positive and gram-negative. In our study, in our previous monitoring study, environmental study, we detected 9 microgram per liter of ciprofloxacin in river water. We detected 9 microgram per liter. So as in regulatory toxicology, we tried to multiply that as a dose response study. We tried to divide that times 10, times 100, times 1,000 as a dose response study. So we used this kind of range of concentration as a dose response study to try to see in which range this amount of antibiotics we detected from the water sample, is it going to cause the imperative defect in Xenobacter elegans. So we used N2 wild type C. elegans. We used the P50 as a food source. We used the NGM plates seeded with, so we seeded the NGM plates with the compounds, and then we exposed age synchronized embryos to ciprofloxacin to the seeded plates, and then we incubated for 48 hours up to L4 stage. So when we reached the L4 stage, we went for the observation by just using different techniques. We used the Lake software for capturing different images. We used the ImageJ software for quantification studies. We used the Nile red staining process during the lipid metabolism study, and the fluorescence microscope and the company microscope just for quantification of different images of our organism. So why we chose C. elegans to study the behavior and the other metabolic or physiological impact of antibiotics, especially ciprofloxacin. We chose C. elegans due to different reasons, but the main reasons here, we said it has a short lifespan. Like within a few days, you will get a good number of embryos, and at one stage, which can be subsequently subjected to tox scans, and then you can have your effect observed within a short period of time. It is genetic stability. We say it has a stable genetic, genomic organization. We have it has a conserved biological pathways. It has a transparent body. Even to some other studies like pharyngeal pumping, you can easily observe its internal orientation, internal organ orientation, like pharyngeal pumping and some other activities which are going on within the body by just observing it easily through the microscope, which is an advantage. We say it has a minimum ethical considerations, so there is no rigorous ethical clearance you need to go through to use this particular animal for your study. It has a simplified nervous system, and we say it has high reproductive potential. So, like one animal can, of a C. elegans can give you a good number of other embryos or at one stage, which can be used in your study. So theoretically, this is what we did in our study. This is what we did. Initially, we did monitoring the study. We detected ciprofloxacin. We detected ciprofloxacin from the river, which was 9 microgram per liter. This was the maximum concentration of ciprofloxacin detected. So we took that concentration into consideration for our monitoring, for our toxicity study. So we subjected the C. elegans to this particular concentration of ciprofloxacin detected from the environment, and specifically water matrix, and then we had to observe different parameters, but the major ones here are behavior toxicity, body morphology, and the lipid metabolism. So we tried to monitor the progress of this animal from its early stages. From L1 stage, we observed L2 stage, L3 stage, up to L4 stage before going into adult. So all of these things we try to observe the progress in growth when it came to body morphology. So we could measure the body length, and then we detected the reproductive age and the reproductive potential of these C. elegans. And then later we used the fluorescence microscope to determine the lipid deposition within the animal after being incubated for 48 hours at L4 stage. So mostly all the parameters were measured at L4 stage. Results. So these are the summary of our results we obtained from our study. The first parameter was to determine to study the developmental toxicity due to ciprofloxacin in C. elegans. So we in our study, we found that when we subjected ciprofloxacin of different concentration, 0.09, 0.0009, 0.009, which is times 10 times 100 times 1000, we found that at both highest and the lowest concentration we used in our study, there was a decline in the body length and the body morphology in C. elegans. So if we could observe here at L4 stage, so at L4 stage, if you can see here, this is the control group. This is a vehicle control, which was DMSO. And then we have 0.009, which is the highest concentration. At L4 stage, the body morphology of C. elegans was declined. It could not even attain the maximum body length which is required at L4 stage. It remained the body length was unaltered, so the body morphology was altered like 100%. It didn't grow to maximum at 0.09, which was the highest concentration. So the next one, and this is the lowest concentration. So we found that in our response study, we found that at L4 stage, especially at the highest concentration, the body morphology of C. elegans, which is shown here, it was highly affected at 0.09. And at 0.09, which was the lowest concentration, still we could get the significant difference in concentration in the body length. So we in this study now, we selected the two concentration, which was a lowest concentration because it has shown a significant difference. And then we selected the next, which was the next lowest concentration, which was 0.09. So we proceeded with these two concentrations for further investigation of other parameters. We dumped the 0.09 because there was an extreme decline in the body length. We could not go further with this concentration. We just dropped it. So we went further with these two concentrations, the lowest and the next lowest concentration for other parameters. So the first parameter was this is one of the behavior for the study of C. elegans, which is feeding behavior. In feeding behavior, practically, we used to count the pharyngeal pumping within the C. elegans, the number of pharyngeal pumping in C. elegans. So what we observed during pharyngeal pumping and the pharyngeal, so this feeding behavior we detected by using pharyngeal pumping, and we found that at the highest concentration we studied, there was a different decline in the feeding habit of C. elegans, and hence we concluded that this concentration highly impacted the C. elegans feeding activities by just interfering. Maybe we are just saying maybe by interfering with the neuromuscular junction, which are highly involved with the pharyngeal pumping in C. elegans, and other maybe some of the toxicity activities which might have taken place, but especially in the neuromuscular junction. So, and the feeding habit of C. elegans just declined. We could not even see like the animals trying to take any kind of food, and therefore we concluded that at this highest concentration, studied concentration, the feeding habit of C. elegans was declined at this much of concentration, which is 0.09. But at the lowest concentration, we could not find any significant results for this. So 0.09, which was the lowest concentration, did not impose any toxicological effect in this animal. So this can be easily viewed by just using this pictorial presentation. We say an elevated amount of ciprofloxacin, it reduced the feeding activity of C. elegans, the elevated amount, which was 0.009, this one. So if you could see the control group here, you can see the pharyngeal pumping activity taking place. The pharyngeal activity is taking place. It can easily be seen like the animal is taking food. Similarly to DMSO, if you can observe carefully, there is pharyngeal pumping seen and the animal is trying to take food. But for highest concentration, the animals was struggling to take food and they would not even easily see the pharyngeal pumping. lowest concentration, the pharyngeal pumping was seen taking place, but the food intake was poor. But there was no significant difference in our results of at the lowest concentration. So we concluded that at the highest concentration or elevated amount of ciprofloxacin, it reduces the feeding activity due to different reasons. One of them being the interference with the neuromuscular junction or mitochondrial dysfunction. It means the animal lacked the energy for pharyngeal pumping activities, and hence there was a poor food intake. So the maximum number of animals used per group was 15, and the magnification was at 8x. So another behavioral activities we try to observe, the behavior toxicity we try to observe in this animal was thrashing activities, and one no-nano response. The thrashing activity, this is the locomotion, is one of the locomotion behavior in cell organs, whereby the animals, when it's exposed to M9 buffer, instead of crawling, it will be having this thrashing activity. We say it's the swimming activity of a cell organ when it is exposed to M9 buffer, just like swimming activity. But when it is on the normal NGM plate, normally this animal will be crawling. But when it comes to the swimming, when it comes to M9 buffer, it will be just swimming. And this we term it as a thrashing activity. So we determine the locomotion behavior in cell organs through thrashing activities in this M9 buffer. So what it does, what we obtained at the highest concentration, there was a decline, extreme decline in the thrashing behavior of cell organs, and at the lowest concentration also, still we had a significant difference in the thrashing activity. So both concentration, lowest and the next and the highest, both of them impacted the thrashing or locomotion behavior in cell organs. Then we went to another activity, which is one no-nano response. One no-nano response. One no-nano, this is the chemical with a very strong smell. So once it comes nearby the snout of the cell organs have to move away from this chemical. So what specifically this one no-nano response measures, it measures the destruction of a neurotransmitter within the cell organs. So when there is a damage or there is any impairment of a neurotransmitter in cell organs or any animal, this one no-nano response will be poor in cell organs if there was any damage. So what we obtained from the study, when we pressed the one no-nano response for the cell organs, we could not find any significant difference among the highest and lowest concentration. So the animals, we concluded that in our study, the neurotransmitters in cell organs was not affected when these animals were subjected to ciprofloxacin studied concentration, which is 0.009 and 0.0009 milligram per ml. So both of the concentration, there was no significant difference in the response towards one no-nano. This is called one no-nano assay. So we concluded there was no any damage or destruction in the neurotransmitter or receptors in the cell organs towards the one no-nano chemicals. So ciprofloxacin could not affect significantly the neurotransmitter in this animal. So this is the diagrammatic. This is the video presentation of a thrashing behavior, which is a locomotion, one of the locomotion assay in cell organs. So we could see the strength of thrashing activity. Through this strength, we will just determine whether these animals were affected or not. So we can see the swimming activities taking place. The swimming activities in the OP50, which is a control group, it is taking place very strongly. Animals are able to swim with energy. They have energy to swim. Similarly to the vehicle control, this is the vehicle control. Animals are swimming strongly and they have good energy. You can see they have good energy for swimming. But here, in the highest concentration, animals are swimming, okay, but their rate of swimming is very sluggish, is very slow. So we concluded that at this concentration, the swimming activities or the thrashing activities of C. elegans decline at the highest concentration. And for this lowest concentration, the swimming activity is okay. It's sluggish, but it's not significant. You know, give significant results. So when we found, when we compared the vehicle control and the control group with this highest concentration, we found that there was a significant difference in the swimming activities between the control group and the highest concentration. So we concluded that the thrashing behavior or the locomotion behavior in C. elegans was affected at this highest studied concentration of ciprofloxacin. Another study, so this was another study we did, the impact of ciprofloxacin on lipid deposition in C. elegans. So this lipid deposition study is being done by a Nairi red staining procedure. So here we determined this study will be determining the lipid metabolism of C. elegans when exposed to ciprofloxacin. So we try to determine how ciprofloxacin is affecting lipid deposition or metabolism, overall metabolic activities. So specifically, what is done, we just stain the L4 stage worms, and then we go to the rhodamine filter, and then we try to capture the images, and then we use ImageJ software to quantify the amount of lipids present in these particular animals. So what we obtained, surprisingly, we found that at the highest concentration, which was 0.0009, there was no any significant difference in the lipid deposition in C. elegans. But at the highest concentration, we found there was a significant lipid deposition in C. elegans. It means there was more deposition of lipids within the worm at the lowest standard concentration. So there are various studies which have accompanied this kind of study, which say that maybe this concentration, this much concentration, is estimating is encouraging the deposition of fat of lipids within the worms by just interfering enzyme activities like lipase or acetyl enzymes which are involved in lipid metabolism. So we found that maybe these enzymes are just this ciprofloxacin is interfering with lipid metabolism. Hence, the metabolic activities around lipid is very poor. Hence, these lipids, instead of being degraded or being metabolized to give energy to the animal, being incorporated into other metabolic activities which are taking place within the animal, is very poor. Hence, these lipids are being deposited into the animal instead of being metabolized for various activities within the animal, due to the interference of this ciprofloxacin. Another study, which is reproductive behavior. So we studied the impact of ciprofloxacin on the reproductive potential for health in C. elegans. So in this study, we raised the worms. We raised the worms from embryos to L4 stage, but these worms were treated with toxicant or ciprofloxacin from the embryonic stage. So when we reached the L4 stage, we transferred like 10 worms from original plates and then to another place, and then started observing the laying of eggs, the behavior of laying eggs of each particular animals in each group. So here we could count the number of embryos which are being laid every day on each particular animals. So we found that at highest concentration there was a decline in the number of embryos or brood size which were being laid by the parents. So there was a definite decline in the brood size at this highest concentration. But at the lowest concentration, the brood size was not significantly affected, this group. So then we went to study the reproductive age. So this, as we transferred the parent animals from one plate to another and counting the brood size, we found that the reproductive age at highest concentration, at the highest concentration, first of all, at the lowest concentration, it was not affected. It went up to 5.5 days or to maximum 6 days. We could still observe animals still laying more embryos, still giving more embryos. But, and this is given in purple color, but in highest concentration, the embryo laying behavior ended up at the fourth day. Fourth day, we could not get any embryos being laid by this animal, as it is seen here on the fourth day. The reproductive age was maximum 4.5 days. But for highest concentration, it went up to 60 days. We still observed the embryos still being laid by these parent animals. So we said that we concluded that at this concentration, there was a maximum reproductive age of 4 days. So at the fourth day, everything ceased. The animal could not lay more eggs. It declined. So what can be the reason behind it? We say maybe this highest concentration could have interfered with the endocrine system of the animals, like those hormones which are involved in the reproductive system of the animals. So hence, there was now cessation in the laying eggs behavior and the overall metabolism of the animals towards reproductive activities. So then we went to quantify the embryonic viability. Embryonic viability, this is the probability or the percentage at which the embryos which have been laid will hatch into L1 stage. Yes, the animals have laid eggs, okay, or embryos. So what is the probability of these embryos to hatch into L1 animals? So this is what we calculated here. The probability of these embryos to hatch into L1. So we found that at the highest concentration, the probability of embryos to hatch into L1 declined. It was significantly affected due to the exposure of ciprofloxacin. Same explanation could be incurred due to the brood size. So we say maybe the interference of the hormones which are involved in reproduction to be one of the reasons encourage the embryos not to hatch. It means the conditions were not favorable for these embryos to hatch into L1 stage. But for highest concentration, there was no significant decline. There was no significant interference in embryonic viability. The viability of the embryos, there was almost 100% of all the embryos laid was hatched into L1 stage at lowest concentration. Conclusion. So what can we conclude from this study? We say the study demonstrates that ciprofloxacin contamination in the environment can exert significant toxicological effects on C. elegans, as we have seen before, parameters, 3 parameters on the behavior toxicity, lipid metabolism, and the behavior toxicity, lipid metabolism, and the reproductive behavior, and the locomotion behavior. So we say this is ciprofloxacin can cause disruptions, as we have seen in growth, where we have seen potential decline in the body morphology. It has interfered in locomotion. It has interfered. We could not see any chemical stimuli response or interference with the neurotransmitter by using one known ASN, but it could have interfered. The feeding behavior of animals has been interfered. Reproductive capacity has been interfered in the lipid metabolism. So this is a great alarm for this much concentration detected from the environment that can pose all of these behavior, toxicity, lipid metabolism has been interfered due to this much concentration which we have studied. So the findings in this study underscores the need for further research to understand the environmental hazards which are caused by ciprofloxacin. Here we are suggesting the studied concentration, relevant environmental concentration which are being studied in different matrix, being soil, being water, being any other form of matrix. Once it gets in contact with other animals, including humans, so here it is reflecting this can be replicated into human beings. So human beings are subjected to the environmental contamination. Hence, this can cause some serious effects in behavior or reproductive or metabolism at different levels, even in human beings. So we are advising like appropriate institution to develop appropriate strategies for the mitigation of environmental risk through supporting legal actions against violators by enhancing the enforcement of environmental protection laws, especially towards disposing of the wastewater or wastewater treatment point from pharmaceutical industries, from hospitals, from the agricultural activities. All of this should be purely treated before being exposed to the water source, which are being said to be the receiver of this wastewater from the wastewater treatment point. So these are some of the basic references we used in our study for conceptualizing the study and getting to our investigation and hence getting our results. Thank you all for your attention. If there is any question, please, you are welcome. Thank you for listening. Thank you so much.
Thank you so much, Mr. Elias, for your excellent presentation. It was very nice and informative as well. And I would like to request, please share the email ID over the chat box. If participants have any queries later on, they can directly email to you.
Okay. Yes, I have shared my email.
Yes. Thank you. Thank you so
in the
much for
chat
saying.
box.
Yeah. This is your official
Oh,
email ID. Okay. So I would like to request
yes.
everyone, please note it down, Mr. Elias present means that email ID. If you have any further queries or any collaboration proposal, you can directly email to him.